In this video, we are mainly talk about DNA replication and translation transcription of the RNA.
DNA, or deoxyribonucleic acid, is a double helix formed by base pairs attached to a sugar phosphate backbone. It is a hereditary material in humans and almost all other organisms. Every cell in a person's body has the same DNA. The location of the most DNA is located in the cell nucleus, but some small amount of DNA can be found in the mitochondria. The information in DNA is stored as a code made up of 4 chemical bases: A, C, G, T. Human DNA consists of about 3 billion bases, and more than 99% of those bases. The order or sequences of these bases determines the information available for building and maintaining an organism. Each base is also attached to a sugar molecule and a phosphate molecule. Together, a base, sugar, and a phosphate are called a nucleotide.
Now, we are talking about DNA replication and the role of the enzyme. The DNA replication is a semi-conservative process. The parallel strands are separate and work as a template for the new strand. In the first step of replication, the helices will unwind the DNA helices by breaking the weak hydrogen bond between the complement bases. The point where they are separate is known as the replication fork. In this process, the single-strand bonding proteins also join to ensure the DNA molecules keep away from each other. Later on, the DNA polymer III read the template to build a leading strand. Meanwhile, the DNA primer also present on the understrand to make RNA primers, which is a starting point for DNA polymer III to build a logging strand. So the logging strand goes in a small section called Ozaki fragment. Then the DNA polymer I remove the RNA primers and replace by a nucleotide. The Ozaki fragments are also joined together by DNA ligands. Then the helices continue to unwind. The leading strands continuously grow toward the replication fork in a direction 3 to 5, and the logging strand discontinuously grow away from the replication fork.
Nucleosome in DNA packing. Histones are used to package the DNA into structures called nucleosomes. Nucleosomes help to supercoil the DNA, resulting in greatly compacted structure that allows for more efficient storage. Supercoiling helps to protect the DNA from damage and also allows chromosomes to be more mobile during mitosis and meiosis. Transcription. Transcription is the process by which a DNA sequence is copied into a complementary RNA sequence by RNA polymerase. The process, the process of transcription can be simply divided into 3 main steps: initiation, aggregation, and termination. At first, in the initiation, RNA polymerase binds to the promoter and causes the unwinding and the separating of the DNA strands. Second, aggregation occurs as the RNA polymerase moves along the coding sequence, synthesizing RNA in a 5' to 3' direction. Thirdly, when the RNA polymerase reaches the terminator, both the enzyme and the recent RNA strands detach and the DNA rewinds.
The translation is a process of porting the synthesis from the information contained in a molecule of a messaging RNA. This process will take place in a structure called the ribosome. The ribosome can be divided in 2 sections, a small and a large subunit. Also, there are complex molecules composed of several ribosome RNA molecules and a number of proteins in it. Translation of RNA molecules have 3 stages: indentation, elongation, and termination. During the indentation, the small ribosome subunits bond to the RNA sequence. Then a transfer RNA molecules carrying the amino acids bound to something called the start content of the mRNA sequence. The start content in all mRNA sequences have the sequence AUG. Later, in the elongation stage, the ribosome continues to translate. Also, each corresponding amino acid is added to the guanine chain, and the peptide bonds are formed between them. This process continues until all the contents are read. Finally, the termination takes place when the ribosome reaches a stop content.